Hi, my name is Lydia Sohn, and I'm from the University of California, Berkeley in the Department of Mechanical Engineering. I'd like to thank the organizers of this conference for inviting me to give a talk on Node-Pore-Sensing, a versatile method to phenotype cells. So my lab is very much interested in developing various modalities to sense breast cancer and to determine susceptibility for a woman to develop breast cancer, which on average, 1 in 8 women will develop breast cancer during her lifetime. One thing that my lab became aware of is a technique called random periareolar fine needle aspiration, or RPFNA. We like to call it the PAP test of the breast, and I certainly know that there are other methods such as just fine needle aspiration. But RPFNA was used and is still used to sort of monitor women who have a known high risk of breast cancer. And what is known is that it will randomly test the breast tissue in various places, and then then you do a cell stain to determine cells that have hyperplasia or whatnot. The problem with RPFNA is that it's a manual process, and that it's known that some women who undergo RPFNA 1 month will look fine, and then months later, suddenly they have developed detected for full-blown metastatic breast cancer. And so the real question is there any way to assess these cells quantitatively so that you don't miss cells, the rogue cells that are in your sample? And this is where my technology comes in, and we always thought that perhaps there's a way to do something called mechanophenotyping cells. That is, to measure the mechanical properties of cells and be able to determine whether or not cells are transforming into cancer or what are normal cells versus cancer cells. So how do we mechanophenotype cells? We use a method that we call mechano-node-pore-sensing, and it really is a very simple 4-terminal measurement of the current to measure the biophysical properties of cells. So in order to really understand mechano-node-pore-sensing, you need to have a little bit of background. And this is the technique that we initially developed, resistive pulse sensing for screening cells. And for those of you who do flow cytometry, you would recognize this nothing more as the Coulter counter principle. So what I show you here is a microfluidic channel that we filled with red dye, but you can see the various parts of this device and this channel. And essentially, you can see there are filters. There's an inlet and outlet reservoir. There is a narrow channel, and then we have 4 electrodes, current and voltage, and with that we measure the current across that channel. So when there's no cell in that channel, this channel is filled with conducting fluid, and you measure some constant current as shown. But then when a cell passes through, that cell is a non-conducting material, or it's a dielectric, and as it passes through that channel, you see a dip in current. Why is there a dip in current? It's really because the cell, because it's an insulator or a dielectric, it's displacing by volume conducting fluid, and so you see less current. And then as the cell completely passes through that channel, you see a rise in current. And the reason being is that the channel once again fills up with conducting fluid. What we've shown and what is done in Coulter counter principle is that the magnitude of a current pulse that's produced by a cell passing through corresponds to the cell size. And in addition, the duration of that pulse corresponds to how long did that cell take to pass all the way through the channel. Now, that is resistive pulse sensing, but what I'm now going to talk to you about is something called Node-Pore-Sensing. So what you see here is just, again, a similar channel, just a straight channel, and what we've done is we filled it with some dye. And then what we've done is also pass through some cells, and a single cell that's passed through, you see that square pulse that is anticipated as a cell passes through. Now, in Node-Pore-Sensing, what you can do is really, just because this channel is like a wire, you can change the geometry of that microfluidic channel. So what you see here is that you have a narrow channel, and then you have a widened region. We call that widened region the node, and then we have a narrow channel again. And what you see is that as the cell passes through, no longer do you just get a plain square pulse, you actually get a pulse that's been subdivided into two. Now, why does that work? And it really becomes this whole idea of a volume to volume ratio. So remember what I said, when a cell passes through a channel, it's displacing by volume a certain amount of conducting fluid. Well, when the cell passes through that narrow part of the channel, it's displacing a certain amount of conducting fluid, and it's a volume to volume ratio. And then when the cell enters into that widened region or the node, it's displacing less volume of conducting fluid at that given instant. And so you see a rise in current. And then again, when the cell passes through that narrow section again, you see a drop in current because it's displacing more of that volume to volume ratio of conducting fluid. And so what we can do is we can actually play around with the microfluidic channel and its geometry. Here what you see is a Fibonacci series of nodes and pores. The pores are really the narrow section of the channel, and that's just a name that we give to it historically. But again, you see basically a Fibonacci series, and you see that the resulting pulse that's created by a cell passing through mirrors that of what we have inputted in the geometry of the channel. So the pulse itself becomes a Fibonacci series. With Node-Pore-Sensing, the really nice part is we always know where a particle is in that channel, and this helps us with very complex situations. So I think for those of you who do a lot of flow cytometry, you can recognize this issue that we do come across with our microfluidic channel. That is, when a cell passes through our device, we see a nice pulse or a series of pulses that correspond to the geometry of the channel. However, when we have multiple cells passing through, we get a coincident event. That is, you see that the pulses all sort of start to combine with one another. In the past, what we have done is, because it was too complex, as it would be with flow cytometry, when you do get coincidence events, we would just remove that signal. However, that really does bias our data. And so what we did was we teamed with Mickey Lustig's group in the electrical engineering department at Berkeley. And he's he really does a lot of different kinds of coding for his own data. And so what we learned from him was something called Barker coding. And Barker coding is something that's used for sonar detection. It's used for Wi-Fi and so forth. And there are 9 unique sequences for Barker coding, and I've just given all 9 of them here. They consists of +1s and -1s. And what's really special, and here are just examples of a Barker 5, again, where you have pluses and a minus, Barker 7, Barker 13. But what's really special about Barker codes is that their autocorrelation is basically perfect. That is, you get a very strong peak when you have autocorrelation, and then you have very small side lobes. And so how can we use Barker coding to tease out these coincidence events? Well, essentially, here's just an example where we have, again, some kind of a complex signal. What we can do is, if we know what our Barker code is in our channel, what we can start doing is to cross-correlate the expected signal for the device with what we have in terms of what is reality. And what you see here is that this is for a Barker 7. We're cross-correlating it. Here's a match. And so we would expect in our cross-correlation data that we would have a peak there. And then we subsequently just cross-correlate everything in that signal. And what you see is that we end up getting 3 matches. That is, there's really 3 cells that are passing through at the same time. And those 3 cells produce that complex signal. The really nice part about Barker code is not only can you tease apart coincidence events, but in addition, you can actually determine the speed of your particle passing through, or your cell passing through the device, just by basically creating a bank, a library of all the expected signals that you would have for different speeds and for that particular Barker code. And this is just an example of some actual data, and you can see that we were able to essentially pick out through our cross correlation the different peaks and the different speeds of that the different cells passing through. And this is just an example of the type of data that we get. And so we see that 3 cells had passed through at different speeds through our device. So with NPS, by basically putting in that spatial and temporal information into the design of our microfluidic channel, we can do real-time detection, we can resolve coincident events, and what's really nice is that we can do multiplexing. That is, we can have multiple devices all running in parallel, and we can actually be able to, because we do this cross correlation, we can detect all the different cells that are passing through, and that really helps in terms of throughput for us. So with that, I'll just go back to Node-Pore-Sensing, and this was done by my graduate student, Junghyun Kim, who graduated a few years ago. And what he did was he took advantage of Node-Pore-Sensing in the sense that we always know where the cell is, and he had this microfluidic channel, which he had segmented with these nodes. But what was particular is that he included something that we call a contraction channel, and you can see that there's this very narrow section in the middle of the device. And with that contraction channel there, what it means is that as the cell passes through, it has to squeeze through in order to traverse the entire length of the channel. And so with that, what happens is that here's just an animation to show you what this cell is doing, but you see that we get what may look like a complex pulse as the cell passes through, but it gives us very specific information. At the first part, it gives us the cell diameter, because remember I said that the pulse magnitude corresponds to the size of the cell. We also get what we call the duration of that pulse as the cell passes through that contraction channel. And the longer the duration, it means that the cell is stiffer. In other words, stiffness and transit time through that contraction channel really tells us how stiff the cell is. A soft cell will pass through more quickly than a stiffer cell. And then we have a series of nodes and pores at the other end of the contraction channel. And what that helps us understand is how well did the cell recover from being compressed. And in the next slide, I'll talk a little bit more about that and show you some real data. But with Node-Pore-Sensing, we can get a variety of information from a pulse. We can get the cell diameter, the stiffness, which is very important, the cell deformation, and the cell recovery time, which is an important part of my story. So just to show you some of the data that we get, the actual data, and also just explain a little bit more about that cell recovery. Here, what I show you is a cell passing through one of our devices. On the upper portion is the expected pulse, and the lower portion is the actual data. And you can see that our data pretty much mirrors what we expect to see. And especially, we really like to see that very long, dramatic pulse in the center, and that corresponds to the cell passing through that contraction channel. For cell recovery, what it means is that the cell has to, essentially, if it recovers, it has to really return back to its original size. And we can see that in terms of how well the last part of the pulses really reflect the first part of the pulses, as you can see. And it sometimes takes the cells a while to get that done, to return back to its original shape. And for this particular device, we were measuring how well the cell recovers within a 40 millisecond time period. And what we find is, as I will show you, that some cells simply don't recover and return back to their original size. And we'll see this again in terms of the data that I'll show you. But I just wanted to be able to show you what the data actually looks like, what we have to process, and also what it means to have cell recovery. So this is data, example data of cells that we mechanically phenotyped. We want to show that we could tell the difference between malignant versus non-malignant cells. These are breast cell lines, MCF7, the malignant cells, and MCF10A, which are the non-malignant cells. And this is a three-dimensional plot of the transit time of the cells passing through the contraction channel. L deform corresponds to the fact that the cells are being squeezed on the sides, so then they elongate. So we want to see how long they elongate. And D corresponds to the actual size of the cell before it's being squeezed. The color code corresponds to the transit time, the recovery time of the cells. And this was done under an applied strain of 0.3. And what you can see is that we very much can tell the difference between the malignant and non-malignant cells. At the same time, you can see that there is an overlap between the two, and that overlap is about 30%, which isn't so great if you really are trying to look for those cells that are cancerous amidst a sea of non-malignant cells. So what we ended up doing is we realized that cells have all different size, obviously, in a population. And if you have two cells that are the same stiffness, but are different sizes, they're still going to have different transit times because a larger cell is just going to take longer to pass through than a smaller cell, again, even though they have the same stiffness. So we did a little bit of dimensional analysis to come up with something that we call a whole cell deformability index. And what that index does is it normalizes all our data with respect to size. So we're really just pulling out what would be the stiffness of a cell. And you can see that this index, which will now be referred to as just WCDI, that WCDI is dimensionless and it's just a number, but it does relate to stiffness. And we've shown that it's inversely proportional to cortical tension or inversely proportional to Young's modulus. So if you see a high WCDI, it means that the cell is soft. If you see a low WCDI, it corresponds to a cell that is stiff. Okay, and so we can apply this WCDI technique of normalizing with respect to size, and those three-dimensional plots get collapsed down into just histograms of size, number of cells per WCDI. And what you see is now, again, MCF7s versus MCF10s. The MCF7s have a higher WCDI because they are softer than MCF7s. And most importantly, you see that little red shade right there between the two distributions. That shade is the overlap, and that now it goes down to about 2%. So we went from 30% overlap to 2% just by normalizing our data with respect to cell size. We still extract out separately the recovery time of these cells. So let me just show you how powerful mechano-MPS and mechanophenotyping can be. We collaborate with Mark Labarge at the City of Hope and his postdoc, Dr. Stefan Hines. And Mark is very much interested in how aging relates to breast cancer. And so within the mammary duct, you have two different types of epithelial cells, myoepithelial cells, which are responsible for ductal contraction, and luminal epithelial cells, which are responsible for milk production. It's really the luminal epithelial cells that are prone to carcinogenesis versus myoepithelial cells. And what we did was we ended up measuring these cells blindly. And Mark gave us some cells that primary cells that were from 19-year-olds and from a 23-year-old. And what you see is our data in the gray. This is with respect to WCDI. And we were able to extract out two different subpopulations in our data. And red corresponds to myoepithelial cells, and the blue corresponds to the luminal epithelial cells. And I think what is really remarkable is that we're doing a mechanical measurement, but with Mark, he would do a flow cytometry, a FACS analysis, and we get exactly the same ratios of MEPs to LEPs, which for me always seems amazing because, again, we're doing a mechanical measurement, he's doing a biochemical measurement, and we get exactly the same results. Here's where we measured women who were either 61 years old or 66 years old. And again, the gray corresponds to our data. And here again, we're able to extract out the subpopulations of MEPs and LEPs. And again, our ratios are identical. Now, Mark and I had always hoped that because as a woman ages, susceptibility to cancer increases, that we thought that we would see a shift in the WCDI. We would see a shift in the stiffness of the MEPs and LEPs, but we didn't see that, which was unfortunate because we were very excited that we would. But the amazing part, what got us very excited, is that in the end, we could tell the difference between the different ages based upon the recovery of the cells. And remember, I said that was a big part of our story. And what we found was that cells that were derived from older women, those cells did not recover from being compressed versus cells that came from younger women. So in the bar graphs that you see here, lighter purple corresponds to cells that simply did not recover as well as the darker purples, which would correspond either to, we group them as instantaneous recovery or transient recovery. So that was very exciting for us. And we've since measured hundreds of cells from, I think, over 20 women of different ages. And what I can say right now is that we absolutely can tell a chronological age difference between these different cells. Our story gets much more complex in the fact that we actually also see differences in the mechanical phenotypes of women who have genetic risk factors such as BRCA1 or BRCA2 mutation. And that, stay tuned, that our paper is being put together about that. But what I want to also sort of spend some time in the remaining minutes of my talk is to talk about what other things that Node-Pore-Sensing can do in terms of cell analysis. So what I've shown you is this mechanophenotyping. We can physically squeeze a cell and detect and determine their, analyze their biophysical properties. What I also want you to see is that Node-Pore-Sensing really allows us to do immunophenotyping label-free of cells. And here's just really how we do it in a very simple nutshell of a schematic. That is, because we can segment our microfluidic channel into different sections by inserting nodes, we can actually functionalize the channel, the glass underneath the PDMS channel, with different antibodies. And those antibodies would correspond to a specific epitope on the surface of the cell. So when the cell passes through, when the cell passes through, the cell can interact with different antibodies if it has that specific surface epitope, and it'll just take longer to pass through those sections versus a control or versus an antibody section that it doesn't have an epitope corresponding to that. And what I mean by slow and what's really going on is that these cells will essentially, they'll transiently bind to the antibody, but they won't stick to the antibody. They won't bind to it because we're running at a high enough flow speed so that the cells just don't stick to the antibodies. And essentially, again, we can look at the transit time, the duration of the subpulses that correspond to the specific antibody sections. And we can then say, if it takes longer than a control section, then we know that that cell has that particular surface epitope. So how do we make this device? In the past, and I'm going through what we did in the past just to show you really how far we've come from these initial results. We used to essentially prefabricate our electrodes. Then we would have a temporary PDMS set of channels that we would essentially do our functionalization that would occur with the process that we're using. It would occur over a couple of days under low refrigerated temperatures. And then we would actually have our PDMS channels that would have the actual node pore, which we would then align on top of our completed functionalized channels. And we would actually mechanically bind the PDMS channel onto our glass slide to do our experiments. So very arduous, but in the end, it was very neat to see that we could run cells through our device. Here's just an example of cells NB4 and AP1060. They're APL cell lines. And we tested and we functionalized the channels with CD 15, 14, 33, and so forth. And we looked at how long the cells took to pass through these different sections of our device. So in the right corner, upper right corner, labeled D, this is what we got in terms of the percentage of cells that show these different markers using flow cytometry. And in C is actually our data. And you can see it looks completely different if you were to do, for example, flow cytometry. We're not sort of gating anything. We're looking at individual cells. We're comparing the transit time of the cells in different antibody sections with respect to our IgG control. If the cells are taking longer than the IgG control, then they're positive for that particular marker. And you can see what our distribution is for these different markers and of these cells, and they very much mirror the flow cytometry results. Here's what we did in measuring acute myeloid leukemia bone marrow samples. And this was really a fun one where, as you know, bone marrow samples will have blasts, will have all different types of cell types. And so we really, in some ways, were wondering, like, how would we know which one are the blasts and everything? So the nice part about node-pore sensing is we also get the size of the cell. And so we're able to gate cells that were of a certain size and then look through our data and essentially then be able to identify the blasts and be able to immunophenotype them against CD13, HLA-DR, CD34. And so this was really great that we're able to get samples. We're able to immunophenotype samples. I do want to emphasize that these cells are label free. The label you can think of is actually incorporated into the device, but not on the surface of the cell as you would do with flow cytometry. So this is all pretty great and everything, but as I mentioned, to actually make this type of device is just very painful, as my graduate students would always jump to say, in terms of the length of actually doing the to actually functionalize the antibody onto our glass surfaces, to actually mechanically adhere the PDMS onto the substrate. It was very complicated. So really the question is what to do. Can you make this more of a viable platform? And this is where we use something called high throughput DNA-directed patterning. So what is high throughput DNA-directed patterning? Well, it's shown here in very simple terms, but the way to think of it is we take advantage of hybridization of DNA. And so essentially what we can do is through photolithography, we can make these photoresist templates on top of aldehyde glass slides. And what we can do is with the open regions where glass is exposed, we can conjugate different sequences of DNA oligos onto the surface of the glass slide. And we can do this multiple times. The DNA oligos are always protected. Whatever's been conjugated onto the glass slide has been protected with the photoresist when we create a new template. But then what we can do is we can take antibodies, we can use click chemistry to attach the complementary sequence of the oligos onto the antibodies. And then we can wash these tagged antibodies onto our patterned glass substrate, and hybridization will occur, and the antibodies will be directed and adhere to those specific plates that we've patterned onto our substrate. So here's just an example of these are printed antibodies that we made, and we just wanted to show that we could capture these cells with antibodies that had been conjugated onto the glass surface. Essentially, we were trying to show people that yes, these antibodies are still functioning, even though that they've been adhered to the glass slide through our DNA patterning technique. And so what we did was we patterned EpCAM, anti-EpCAM antibody, and CD2. And essentially we ran JERCAT and MCF cells across our device. And what you could see is that we capture the MCF7s with the EpCAM, and we capture the JERCATs with the CD2. And we get pretty good capture purity, which was pretty nice to see. So with our NPS immunophenotyping, what we now can do is use the same oligo printing technique to print our oligos in specific sections of our Node-Pore Sensing device. You can see that it's slightly changed in geometry, and these are for fluidic reasons. But essentially, we were able to then take antibodies that we had tagged with oligos and essentially wash them across our device, wash them through a completed PDMS device. That's the really nice part. We can prepare everything in terms of oligos, pattern onto our substrate. We can then use our typical oxygen binding to bind the final PDMS device onto our glass slide. And then we can actually just run our tagged antibodies through our channel. They will hybridize into the specific sections of the device. And then we can just wash through with buffer. And then we can run our device through it. So you can see it's a very, very simple method. And I should say that instead of days to complete functionalization, what we find is that we can actually do our hybridization. We can do everything within a half a day. And essentially here, this is just an example to show that we able to show that we can see that MCF7 cells take longer to pass through the EpCAM antibody region versus the control, and the JERCACs don't interact with the EpCAM at all. So what does the MPS platform look like? This is just an example. We built one for the Labarge group at the City of Hope. It was really nice to see that we were able to transfer our technology over. And you can see that it's a very simple platform. Just all that requires are just a couple of voltage and current sensors and a computer. And you can see our little box where Stefan, who's the postdoc who uses it, just has to snap in a device and do his measurements. So right now what we're doing is we're currently combining mechanophenotyping and immunophenotyping in our device. And I just want to end with really, where does our mechanophenotyping, our Node-Pore-Sensing really sort of lie with all in the space where there's quite a number of other methods to do mechanophenotyping. And you can see here we have high resolution, we have fairly good throughput, not as well as flow cytometry or microfluidic cytometry, but because we're running at a moderate throughput, we are able to see the recovery time of these cells very easily. So we just hit a sweet spot. And we're certainly faster than atomic force microscopy and micropipette aspirations, which are really the gold standards for mechanophenotyping. And with this, I would like to just acknowledge my group. In gold are the students who've been very much involved with Node-Pore-Sensing or Node-Pore-Sensing in general, and all my collaborators and my funding. Thank you very much.